just wanted to thank you, Athena, for setting this all up. It's been a fabulous kind of thing to be able to see week by week, not only the different topics and there's sort of different sets of conclusions and data, but just the people involved. It's really reassuring, and I don't know, it's just great to have so many people using these tools around the Bay Area, doing all kinds of different things with all kinds of different critters. So thank you for that. I really wouldn't have known, you know, half the people on this site if it hadn't been for this. So I really appreciate that in particular. From where we are, this is where we work. This is the Hopkins Marine Station in Monterey, just right next to the Monterey Bay Aquarium. It's been part of Stanford University since 1892, and it's the place where we have been launching this work on using community and ecological DNA to study the diversity of kelp forests. And this is all work that has been spearheaded and really headed up by Peter Schrum, who's I saw joined just a few minutes ago. Peter, if you wouldn't mind just unmuting your microphone and giving a shout out from Newcastle.
Hey guys, yeah. Look forward to the talk, Steve.
Yeah, Peter left lab for Newcastle just before the shutdown happened, so he's been there since. This shows the extent of kelp forests and laminarian forests around the world. These are brown algae. They live in shallow waters along coastlines, mostly in temperate coastlines. We have giant kelp, which is the purple line here on the North American, South American coast. But the other major kind of kelp forests are shorter laminarian forests that are shown in the blue here. And you know, the interesting thing about this is that the extent of these forests around coastlines is actually as great or greater than coral reefs around the world. There are also areas of high productivity like reefs. They have a huge amount of diversity like reefs, and so they are another one of the kind of coastal communities that makes up a lot of the productivity and diversity of ecosystems. What we wanted to do was to develop a method to use environmental DNA and ecological DNA to look at the diversity of kelp forests and up and down the California coast and around these areas. And we did that with an initial sampling of around Monterey Bay. The left is another aerial of the Peninsula of China Point, and the yellow dots are the sampling places, and the map on the right just shows you where Monterey is in case you haven't been there yet. This data set is based upon 116 cobbles, typical ecological DNA. And by ecological DNA, I mean that we sampled the cobbles, we had the organisms in our hands, unlike environmental DNA where you might be taking water samples or soil samples. So we have DNA from the cobbles, taken all of it, all creatures great and small. We amplified the cytochrome oxidase 1 gene in all of them, and then can look at the distribution of species and taxa across them. And this is a sort of typical way that people have been using ecological and environmental DNA to look at diversity. I mean, we found a huge amount of diversity in these things. We found well over 900 species on each cobble. They're not all identifiable. Only about 366 are identifiable to family, and about a third of that are identifiable to species. So we have a lot of identified species, but only a fraction of the species that are there are identifiable. That's a whole other topic to be talked about, but that's kind of where this is. There are lots of diversity of small things, lots of the diversity of really big things that leave a track on there. But what I'm going to talk about instead of the diversity at the species level is something that Peter and I have been working on for a bit now, and that the data themselves include not only species level information, but they also include the haplotypes that come from those species. And those haplotypes are in the data set. They're in all environmental and ecological data set, those haplotypes. So for example, in our data set from 527 species that are pretty well known. We sort of take a subset of all the ones that are there across 19 classes. We found out of those 527 species, over 2,500 CO1 haplotypes. A lot of species only have one haplotype. That's on the graph on the lower right. And but a lot of them have multiple haplotypes, and so we can begin to then use the haplotype data to begin to look at other patterns of important ecological and genetic features of these species. So that's what this talk really is about. I'm just presenting essentially the ability to find haplotype data within your environmental or ecological DNA data sets, how you use them to advance looking at ecological patterns or genetic patterns, and then I want to sort of spark a conversation about what some of the nuances of that are. This shows a distribution of genetic diversity, that's pi along the x-axis, across all the 19 different classes that we're talking about. A lot of individual species within all of these classes have zero genetic diversity. They only have one haplotype in our data set, and that would be the histogram on the far left here. And then all the other histograms represent species that have a reasonable amount or a high amount of genetic diversity in our data set. You can look at some of them. For example, the top there is the class Anthozoans that has practically no genetic diversity in our data set. And that reflects a really short or very slow amount of mutation in the mitochondrial DNA of anthozoans like sea anemones and corals. Whereas you go down the list there to the malacostricans, see if I can use my pointer right there. These are small crustaceans, isopods and amphipods. They have a huge amount of genetic diversity in our data set that can be really, really useful. So low genetic diversity in anthozoans and some starfish. What can you use the genetic diversity for? You can do things like this. You can find the ecological aggregation of individuals across samples. So what's shown on these diagrams are a set of cobbles along the x-axis, and then the number of haplotypes found on those cobbles along the y-axis. For the top is a coprillid amphipod, and then on the bottom is a different kind of amphipod. What you can see of the coprillid at the top is that there's 4 cobbles that have a huge number of haplotypes on them. Now, different haplotypes on that cobble mean there's different individuals there, right? They have different genetic signatures. And so because you can count different haplotypes as an index of different individuals, you can use haplotype number as an index of population size. It's not perfectly population size, but it gives you a better index than, say, just the species level reads would. So these coprillid amphipods are hugely aggregated across these cobbles. 4 of those cobbles make up about 90% of the total population that was in our sample. The same is true of the amphipods on the bottom. Across all of the 527 species, about 30% of them are clumped on the basis of cobble by cobble. So this is a good tool to look for aggregation and clumping across cobbles. You can also use that same information to look at population density across different regions. We had 3 different regions, Hopkins and Lover's Point and Point Pinos. And by using the individual haplotypes as an index of population size, we can say, well, how many haplotypes per cobble were there at Hopkins, Lover's Points, and Point Pinos? We do that and find that the number of haplotypes per cobble and the inferred population size for this one barnacle is a lot lower at Lover's Point than Hopkins and Point Pinos. Again, I'm not saying this is a big deal in terms of the diversity of the kelp forest, but it gives you a tool to be able to look at ecological distinctions over very small spatial scales that otherwise is very difficult for ecological DNA to do. So I'm 10 minutes into this, and I'm just going to make sure that there's somebody else there. So somebody unmute their microphone and say, yes, we're here, like.
We hear you.
Okay, good. You just don't really know, right? So you can look at taxa with different abundances. across different groups. We see that in barnacles and starfish and crustaceans and bryozoans and algae, again, as a tool to look at the ecology over small spatial scales. So you can also find population genetic structure in the data set. Once you have the haplotypes and you know their distribution over space, you can ask, are those haplotypes distributed evenly and randomly with respect to space? And you basically calculate an FST based upon the haplotype data. So this is a histogram of the frequency distribution of different FSTs. You see most of them are pretty small, less than 0.01, but a good number of them are in the 0.02 and the 0.03 range, which is pretty high considering that these locations were sampled, they're all about 5 kilometers from one another, and these are marine organisms. So having high amounts of FST over very short spatial scales is a bit unusual. It suggests that there are hidden features of genetic diversity in these data, and we didn't really know that much about. When we dig into what species show high FST, that's what's shown here in this pie diagram, with all of these strange names on the right, like the Floridiophyceae. That's something that you probably will hear ever said again all day. Those are red algae. Red algae don't have a high dispersal phase. They have actually no motile sperm even, and so they have very low dispersal potential. The second one down are the demosponges. There's sponges that have crawlaway larvae. Malacostricans are the gray zone there, and they largely have brooded embryos that crawl away. So that whole pie diagram is dominated here by organisms that we know from the life history have relatively low dispersal potential, and they're the ones with high FSTs across these short spatial scales. So that makes perfect sense again, and it suggests that we can use the haplotypes in order to get an index of population differentiation over small spatial scales. There are nuances here, and these are things that Peter is particularly good at figuring out. For example, there are sequencing errors in all these data, and there are ways of dealing with that. There's a denoising parameter called alpha in most data pipelines. As you bring up alpha from 1 to 10, then you're accepting more rare sequences as being real in the data set. And so what we did, in fact, was to use one haplotype, of this case a conch, and we knew it was only one haplotype. Run that through the denoising algorithm. That's the greenish bluish set of points there. And as alpha goes up, we see one haplotype when alpha is 1, one haplotype when alpha is 2, one haplotype when alpha is 5. We see 30 haplotypes when alpha is 6, etc. So we set alpha at 5 to accept as many of the rare sequences possible without getting too many. But there's also other ways of filtering the data. Essentially, if a haplotype only occurs once in one single read in your entire data set, it's got a high likelihood of error. If it occurs only in one sample, but with more than one haplotype read, there's a moderate chance of error. If a haplotype occurs on 2 or more cobbles, it's very likely to be real. So as you can basically take your data, filter them in these different ways, and get a different index of how of whether or not the noise in the data set is affecting your results. This just shows that for just general genetic diversity, these don't make that much difference because the rare haplotypes don't impinge very much on it. But this shows a distribution of Tajima's D, which is a tree-based estimate of the history of populations, and that is pretty sensitive to the denoising assumption and the rare reads phenomenon. So I'm going to run through this very quickly. Our next data set is 5 times as many cobbles all across California, and that's basically what we're trying to work on right now at the level of species and at the level of haplotypes and population structure. So there's lots of weird things out there in the ocean. We can find them with ecological and environmental DNA. We could also look at the nuances of their genetic diversity. And that's what I had to share with you.
Thank you for the great talk. Anybody have questions for Steve? You can either type a question mark in the chat or type in the whole question. Henrik.
Not hearing you, Henrik.
Unmute yourself, Henrik.
Still not hearing.
Susan to the rescue.
I figured it out now. Sorry, I lost my own screen. So thanks for the presentation. Very interesting, also for what I'm doing. I'm working on a similar project, and I was also thinking about doing this kind of haplotype calling. And one big issue I have with arthropods is that they have a huge amount of nums. And have you thought about that? Is there a way of like safely removing nums from these data sets?
So nums to everybody is basically nuclear pseudogenes, right? That's what you mean?
Yes, that's what I mean.
Yeah. So they mostly, in my experience anyway, yours might be different, they are so different than the normal haplotype that they are not clustered together in the same OTU. And the way the pipeline works is first you cluster together sequences at the 98 or 97% level in OTUs, like usual. And then within those sequences that fall into that OTU, you then break those up into different haplotypes.
So that makes sense, and this is how I initially thought about this as well. So a couple of weeks I reviewed a paper where they suggested some method to remove it, and in that paper they explicitly mentioned that nums can, I mean, very recent nums can actually fall into the same OTU. And they suggested a method which I was not sure is the right one, but I thought maybe you had another idea of these. So with these closely related ones, you wouldn't know either.
Yeah, if they're really, really recent nums, then they may be practically identical to the things that are there. And I don't know what you can really do about that. We don't see that many of them. What we have done though for the data set that is in this submission that we have out is we've taken the set of haplotypes that are really diverse. So for example, we have some species that have over 100 haplotypes. That just seems so weird, and it's just like too many. So we took them all and translated them, and then looked at the patterns of amino acid conservation and mutations, and they act like normal mitochondrial genes. They are dominated by transitions at silent sites. And so under those circumstances, we're sort of accepting them as normal mitochondrial data.
Okay.
Another thing you can do is to take the collection of haplotypes and see whether there aren't any that are following different mutational rules than mitochondria usually do. That'd be my suggestion.
Yeah, thank you.
Great. And Lorraine has a question.
Yes. Hi, Steve. It's Lori.
Hey.
I really enjoyed when you talked about the nuances and how that slide when you showed the different filtering levels of like high probability area, medium probability area, and then probably real. So I want to talk about that probably real because you base that on finding a haplotype on at least 2 cobbles, which makes me think that if you want this method to be widely applicable, then you have to worry about the distance of your different samples, right? Because depending on the density that you sampled the cobbles, that's going to change that third parameter. So I was wondering if you could discuss more on that.
Sure. I mean, it certainly does change what it is, but our thinking was that if you have independent samples, and that you see a haplotype on those 2 independent samples, that means you've got independent DNA isolation, independent amplifications, independent sequencing. And it's really going to be uncommon for sequence error to generate the same 2 haplotypes in those independent ways. It's going to happen sometimes, but it's not going to happen that often. And so I don't think it matters that much that they're close or far. I do think it matters that they're totally independent of one another in terms of collection, extraction, and amplification.
Okay, thank you.
We have a question about what a cobble is.
Good. A cobble is a small stone, just like in a street, and kelp forests and laminarian beds are around because they're coastal and high wave energy areas. Often have cobbles the size of your fist, or up to cantaloupe size, or maybe even a little bigger. And because they are loose, they're easy to collect. You just basically pick them up.
All right, thank you, Steve. Okay, next up, our next speaker is Jordana Muir. Jordana is a student at Stanford, and she's calling in from South Africa. She is going to talk about her project on mammal communities there. And we're a little worried about her internet connection, so we have a pre-recorded presentation by her that I'm going to play, but she'll be back to answer your questions live. So, let's see. Can you guys see this?
Yes.
Okay, perfect.
Hello, everyone. This is Jordana calling in from South Africa. I just wanted to thank you all for this awesome platform to be able to share our research on. I'm currently a grad student in the Hadley's lab, and I'm going to share some of my work on molecular ecological network analysis that we are working on to assess ecosystem biodiversity, trophic interactions, and community structure. I would like to say that this is not published as of yet, and is still a work in progress, but I want to share it with everyone here to get feedback and possibly network through this. So, as everyone is well aware of the global threats to biodiversity, we are more critically, and even more critically, is how it disrupts fragile interactions among species, which are the foundation of functioning ecosystems. So my question is, how will these emerging interactions integrate and rewire these ecological networks, and how are we going to measure them? And one way to do that is through, which is the aim of this project, is to explore the use of a bioassessment tool that combines both molecular and network theory innovations to infer the changes in ecosystem biodiversity, trophic interactions, and community structure. So throughout this, I'm going to be calling it MENA, Molecular Ecological Network Analysis, because that's a mouthful, and MENA is quite easy to say. So from the earlier talks, I'm sure metabarcoding was covered extensively, but would like to mention how this technique is enabling accurate and cost-effective biodiversity assessments at a level of precision not previously available. And now we can use this information to construct highly diverse and complex ecological network analysis. And these networks provide a quantifiable approach using species interactions to explore the ecological complexity, the structure, and the resilience of communities while identifying key species. So to assess the robustness of these measurements, we require an area that has been well studied. And we're very fortunate to have our own preserve on campus called Jasper Ridge Biological Preserve, which is located about 45 minutes south of San Francisco, which is where a lot of you are sitting, I believe, right now at Cal Academy. And within this space, the ongoing research has been done since the 70s, so we have a good idea of the biodiversity that's occurring within this space. And just to point out that it is on the fringe of a highly densely populated Silicon Valley. So the advantage of this well-studied system is also the long-term camera trap records that have been going on since 2012. These figures are coming from Kevin Leopold, who is a postdoc in our lab, who has a paper and review on this, but I'm sharing these to show you the importance of the long-term data that we have. So the date is along at the bottom here, and the photo trap rate is on the y-axis, and we're following this black line, which is the trend line over time. And what it did was capture the return of an apex predator and that cascading impact on the other mammals within this system. So we see that in 2013, after the arrival, we have a decline in coyotes and in deer occurrence, and a slight increase in the jackrabbit. And after the decline in coyotes, we see an increase now in foxes, in which they actually didn't occur on Jasper Ridge. And the bobcats were not impacted at all by these shifts. So to better understand these changing dynamics and how the restructuring of the food web is actually impacting this system, we wanted to develop and then a pipeline. So the objectives here today are to determine carnivore, omnivore, and herbivore diet composition through DNA metabarcoding of those 6 species that I showed you on the previous slide. We will then reconstruct the structural and functional relationships within the food web using network theory, and then determine the potential of MENA as a biodiversity assessment tool to infer community diversity and structure in the system. So we collected about 150 fecal samples in 2 short 12-day sampling periods and froze sampled immediately. We used the Zymo Quick DNA fecal kits for extraction. And then we targeted 4 different groups that we were amplifying: mammals, plants, endoparasites, and arthropods. Today, I'll just focus on the mammals and plants within this community. And I would just like to point out that we chose 2 primers that had quite large base pairs, and this was so we could get higher taxonomic resolution within the diet. We use Illumina-MySeq platform, and with that, we're able to get 20 million reads for the mammal primers and about 2 million reads for the plants. And we used OB tools and a very conservative pipeline for the bioinformatics and the filtering process. You'll notice in my question that we lost quite a few reads from 20 million down to 4 million for the mammal primer, and this is because a large proportion, 60 to about 95% of those reads were actually the host DNA. And that's great for identifying where the scats are coming from. However, it does detract from the diet. We then created a database using ecoPCR, using the MBL reference database. And I'd just like to point out, because of this community that we're talking with right now is very interested in this, is questioning, how does the reference database compare to the historical species list? So we're only good at assessing the biodiversity of an area depending on what the database is that we're comparing that to. So at Jasper, there's 762 plants that's previously been recorded, and using this primer and comparing to what is in the database, we were able to identify 45% to the species level, 43 genus. However, about 5% was not in the database, and that's about 40 different species. And then looking at our historical mammals that have ever been recorded on Jasper, including bat species, does a very good job of picking up everything, except you'll notice 4%, sorry, let me go back, 4% to the family group, and the key species being California ground squirrel is not in the database. However, we do know that mammals, our key species, are eating them, but we can't pick this up in the diet. So just interesting note to point out. We then use these taxonomic units to create our ecological network in iGraph and in Gephi as a visualization. So we ended up with 32 carnivore, 57 omnivore, and 25 herbivore species over this collection period. And we're able to identify 212 unique reference sequences. And we did that to almost 90% to the species and genus level, so quite specific. And then if we're being more conservative in our analysis, we combined the reference sequence IDs that were to the same genus or the same family level. So we are left with 167 unique IDs, 91% identified to the species and genus level. And from the short collection period, we can see that we are able to capture a large amount of the species richness, dietary richness of this area. So two measurements that we're using is the frequency of occurrence, which is the average occurrence of food items across the samples, and the relative read abundance, which is the proportion of the unique Illumina sequence reads in a sample divided by the final number of sequence reads in that sample. So just touching on a few key points here, looking at the predator-prey diet, there were 16 different mammals, comprising of 11 families, 13 genera, and we're able to identify 75% of those to the species level. Here, we looked at the functional groups of what was being consumed within the system, and small mammals is anything under 2 kilograms. And we see that the puma and the coyote are eating larger prey versus the bobcat and gray fox, which are smaller mesopredators eating smaller mammals. And we found that to significantly correlate with the body mass of both predators, the predator body mass and the prey body mass. Looking at species overlap, niche overlap of their diets, we would have thought that the puma and the coyote would have overlapped significantly, coming from looking at our camera trap data. However, it's the bobcat and the gray fox that are highly overlapped within their mammal diet. However, this changes when we add plants to the diet of the gray fox. They're supplementing quite a bit of their diet with plants. We found about 78% of the samples contained plants. And 196, we actually identified 196 plant reference sequences representing 106 genera, and that's 44 families. We see here, looking at the relative read abundance of plant functional groups, the coyote and the gray fox both consumed mainly woody vegetation, and diving deeper into what those species were, they were usually fruit-bearing species. And the deer is a mixed feeder between woody vegetation, grasses, and more herbaceous vegetation, and the jackrabbit mostly consuming herbaceous plants. Okay. So now, comparing this as a biodiversity assessment tool, we were able to compare this to the camera trapping at the same time, along with soil eDNA study that was taking place again by Kevin, who has recently published this information. So while he was collecting that data, I was also collecting Scat samples and looking at how this compares to the other two. So as you will note here in the yellow, this is representing what was found in the fecal samples, and you will see that there's a lot of overlap with an extra 7 different species compared to cameras. And this is mainly the small mammal community that's often missed by camera traps. And you'll see the soil eDNA and the soil and fecal both missed as expected, the California ground squirrel, because it was not in our database. Now, when looking at the mammal species list of Jasper Ridge, the more recent one, not including bats, I'd like to point out that the fecal and soil eDNA were identified 73% of those species, and the camera trap only 59%. And then looking at our plant species list of Jasper, we identified our diet was made up, 82% was found on that list and 18% outside. So I'd like to point out that there's that 18%, along with these species here, the house mouse and the wild boar are 2 species not known to ever exist within Jasper. So meaning our animals are consuming a wider breadth than is inside Jasper, and we're able to monitor a larger community across this space through these assessments. And then one thing these other biodiversity measures cannot do is to identify the exact interactions that are taking place and to be able to reconstruct these multi-trophic food web as we have done here. So just to show you what you're looking at, this food web of Jasper Ridge, the nodes of these dots are representing the mammal and the plant OTUs within their diet, broken down to the 106 plant genera. And the colorations are all represented by their functional groups. The size of the nodes are the degree of connectance. So the more connected you are within the community, the larger your node size. And then the edges are your links representing the frequency of consumption. So deer are eating a lot of oak, whereas puma are eating a lot of deer in this situation. So with this network, we are then able to ask questions about the non-random structural patterns of the community that are expected to influence ecosystem stability, such as modularity and nestedness. So within Jasper, we found the community, or this network, is actually more modular than expected by chance. This means that the communities within are more compartmentalized, as we see here in this little diagram, where a collection of nodes, which are more densely interconnected than would be expected. So modularity is a particularly important property because it is related to robustness. So if a species was, say, removed from this modular, it will not have a collapsing impact or a knock-on impact on these other modules. And that's what we consider a more robust system. Nestedness, the other measurement of community structure, is a species assemblages occurs when the specialist species only interact with a subset of the species generalist. So as we see here in this adjacency matrix, the more organized the network is, the more nested it is. So this is nested, representation of nested versus non-nested. And in Jasper, in this case, is a non-nested community where species diets are not strictly overlapping and dependent on one another. And therefore, again, another representation of a system that will be more resilient to outside changes. So we can also be measuring robustness in this community and secondary extinctions. And we have identified key species throughout this system using this techniques, but I won't get into all of that at the moment. But just as a quick recap, this non-invasive metametrics can act as a robust biodiversity and community assessment tool. We see that it is capable of identifying key species within a community, and it also identifies potential changes in and the subsequently of ecosystems in a rapid changing world. So looking forward to taking it to the next steps. I would just like to thank everyone here and all the help that I've had from the lab and undergrads and advisors on this. So thank you, and would love to have any questions from anybody. Thanks.
All right, that was great. Question for Jordana.
Hi, showing that. Sorry, it was still a little degraded there.
Okay. So we have a question. I want to use open source software to compare or get species ID from GenBank, as I saw people are using Genius, but it paid software. Do you have any idea which software I can use instead of Genius to get species ID from GenBank? Oh, I think that question was answered.
Yeah, I think you're right. Yeah.
Any more questions for Jordana right now? Anik?
Unmute myself. Thanks for your talk. It was really interesting. I was curious about you seem to get quite a high percentage of identification from your fecal and soil samples. So I was just curious whether you think that's related to the fact that this system is so well studied, or whether you think that would be replicable in other field sites as well.
That's a very good question. I think having it studied before, we're actually able to compare and know what is actually there in that system. But we did get a high resolution to the species level and the genus level, so that would occur in most situations. But if you are studying, like I am actually working in the Congo in these remote regions, I don't know if the database, of course, is going to be as good and replicable in those situations. So yeah, I mean, even in this great California system that should be well studied, we don't even have ground squirrels in there. So yeah, that's something we need to work on. But people are working towards that and increasing the database and sharing their data. So hopefully we'll be getting better with that.
Yeah, cool, thanks.
Great, and Steve has a question.
Hi, Steve.
Yeah. Hey, Jordana. Nice to see you.
Hey, good to see you.
Great.
Great.
Thank you for recording it, especially given the glitchy nature of the internet world. My question is about measuring the amount of interaction among species and the amount of, say, predation by one species on another. You've got individual scat, and are you taking the abundance of species interactions, just the number of scat that have a particular species in it? In other words, are you just doing presence absence of a species in the scat, or are you trying to use the read density of a species in the scat as some index of how much that individual was eating?
Right, very good question. So I was using the frequency of occurrence so that it did take into account the number of samples that I collected per species, because it did differ. So we used frequency of occurrence for those interactions. And then when we were looking more at the functional groups that were being consumed, we did use the relative read abundance. And kind of comparing both frequency of occurrence to relative read abundance, all of it. Both of those, yeah. Are you guys using frequency of occurrence or relative abundance? Because I know that's a big issue in this topic, like which to use.
It is a big issue. We are only using presence absence as an index of abundance, because especially when you've got individuals that are really small, like the copepod, that could be really big, like the starfish, it makes a big difference. But the other aspect of using the haplotypes is that every time you have another haplotype, say in a scat, that was 2 individuals that were eaten, and so you can count haplotypes. Presence absence of haplotypes gives you a higher level of certainty of what the population size is in the interaction. So, we're all doing the same thing. Like, how do you count things?
Yes, no, exactly. But yeah, I would love to talk to you more about the haplotypes and seeing if that's actually possible within what we're doing. I'm not sure.
Rosie.
Yeah, thanks. It was a great talk, Jordana. And I mean, we're doing very similar things with arthropods to try and reconstruct food webs, and so kind of reconstruct the whole community. But I was trying to figure out from what you said, were you able to actually say that these animals were eating plants versus eating the animals that are eating the plants? In other words, were you able to kind of know that these were actually feeding on plants rather than just you're picking up the contents of the diet of the
other one? Yeah, that is a fantastic question, and I'm so happy that you answered it, because I didn't really want to go down that road. But that's things with the fox. Are we picking up the diet of the rabbits that it's eating? And we did look at. I mean, there's no way to tease that out, unfortunately. Except maybe looking at relative read abundance and things that are of low abundance that match with the rabbit. Maybe those are secondary consumptions. But what we can see is that there is general overlap in a lot of the samples. But then I do have, what did I have? About 40% of the fox samples that were eating mammal preys, but did not have plants in those diets. So that's a really good question, and something we're trying to figure out how to tease it apart. So if anybody else out there has suggestions on this, that's a really tough one and a good question.
That's
yeah.